Hello, and welcome to Effective Means to Transfect and Immuno-precipitate GFP-Fusion Constructs, presented by BioCompare and sponsored by Bulldog Bio. My name is Mike Okimoto. I'm the editor-in-chief for BioCompare, and I will be today's moderator. Before we begin, I'd like to remind our viewers that this event will hold a live question and answer session at the end of the presentations. You can submit a question at any time using the Q&A box located on your screen. Also, please note the resource box containing downloadable documents pertaining to this webinar. Okay, so allow me to introduce today's first presenter. Having performed more than 500 immunoprecipitation experiments during her scientific career, Dr. Tina Romer is now the head of biochemical research at Chromatech and responsible for developing new tools making IPs easier. Chromatech GmbH pioneers a new class of immunologic research tools derived from single-domain camel antibodies for cell biology and proteomics. Welcome, Dr. Romer.
So thank you, Mike, for the kind introduction, and now I welcome everybody to the first part of the webinar, Troubleshooting Immunoprecipitation. Well, I think even if you're familiar with immunoprecipitation, it makes sense if we first get a general overview of the different steps of an immunoprecipitation reaction before we start with the troubleshooting. If you do an IP, you want to isolate your protein from a cell lysate using an antibody coupled to a solid matrix. And probably the most important step in your IP is to choose the right antibody. Because if your antibody is not working, your whole IP cannot work. So first you choose an antibody, and then you couple your antibody to a matrix. As matrix, often beads are used, or sometimes also, for example, 96-well plates. After choosing an antibody, you have to prepare your cell lysate. Cell lysates can be prepared from many organisms, like eukaryotic cells, plants, yeast. It depends which organism you are interested in. To start your IP, you then add your lysate to the antibody coupled to the beads. After incubation, you wash away all unspecific bound proteins. And finally, you can elute your protein of interest from the beads. Your eluted protein of interest you can then use for all kinds of downstream applications. So the principle of an IP reaction sounds very simple and straightforward. However, all of you who already did an IP know that there can be many pitfalls during such an IP. So now let's start to have a closer look at the single steps. As I said before, if you start your IP, you have to choose an antibody against your protein of interest. And please remember, not all antibodies are working in IP. Frequent problems that might occur is that your antibody binds unspecific to many proteins. Then you will have a really high background afterwards in your immunoprecipitation reaction. Or it might happen that your antibody doesn't bind at all or only weak to your protein of interest. Then you won't have any immunoprecipitation. And please also remember there are antibodies which are very well working in Western blot, but this doesn't mean that they immediately also work for IP, because it might be the case that this antibody only recognizes denatured proteins, and during your IP you normally want to have your protein in a non-denatured form. So to avoid these problems, you can use antibodies that are validated in IP, that are published in IP experiments, You can use antibodies that were recommended to you, or you can also see whether you can find an antibody which is certified by the antibody company. And if no good antibodies are available, think about fusing your protein to a protein tag. Good antibodies are available against protein tags like HA, MEK, FLAG, or GFP. After choosing your antibody, you have to choose your matrix. So often, antibodies are coupled to protein A or G beads, since many antibodies are bound by protein A or G. Using protein A or G beads, you are very flexible. But if you then want to elute your protein, you have to remember that you not only elute your protein of interest from the beads, but also the antibody. And depending on your downstream application, the heavy and light chains of the antibody can be disturbing. To reduce heavy and light chain contaminations in your downstream application, you can cross-link your antibody to protein A or G beads, or couple your antibody directly to activated beads. Using this system, you are still very flexible, but it is more time-consuming and needs more validation. A third option is to use beads that are ready to use. So you just take them out of the fridge, wash the beads, and start your IP. This system is very fast, and since the antibody is cross-linked to the beads, you again have reduced or even no heavy and light chain contamination in your downstream application. The disadvantage of ready-to-use beads is the limited choice of antigens. Ready-to-use beads are probably not available against all proteins you are interested in. After choosing the antibody and coupling it to a matrix, you need to prepare your cell lysate. During lysis, you want to solubilize your protein, otherwise you can't do an IP. Ideally, your protein is kept in its native conformation, and for sure you don't want to have protein degradation. And you also don't want to lose your post-translational modifications if you're interested in. At least for eukaryotic cells, mainly two buffers are used which solubilize nearly all proteins. I know that there are more difficult proteins like membrane proteins, but discussing solubilization and immunoprecipitation of membrane proteins would definitely extend the time of the webinar. But as I said, for many proteins, mainly two buffers are used. The first buffer is a non-denaturing buffer. It contains tritron or NP40. This buffer is very suitable if you want to do co-immunoprecipitations, since it does not denature your protein, and during the IP you also then won't lose your interaction partners of the protein. But you might have problems to solubilize some proteins like nuclear protein using non-denaturing buffers. To solubilize these proteins, you can use REPA buffer. However, REPA buffer is slightly denaturing, and you might have problems to solubilize some proteins like cell-like nuclear proteins. But you might have problems to do a co-IP if you use repair buffer. And again, please don't forget to add protease inhibitors or other inhibitors like phosphatase inhibitors, depending on your protein and your interests. Well, after preparation of the lysate and the choice of the antibody, you can start your IP reaction by adding the antibody to your lysate. As I mentioned before, you need a good antibody to catch your protein from the lysate. Otherwise, your IP won't be successful. But you also might have to optimize several other parameters. So during your actual immunoprecipitation, you can play around with the salt and detergent concentrations. And you can also try to adjust time and volume of your immunoprecipitation reaction. The same is true for the washing step. During the wash, you can also optimize salt and detergent concentrations and time and volume. Elution of the protein from the beads is often done with denaturing SDS sample buffer. But if you need your protein non-denatured, you can try to elute with a glycine pH 2.5. But please keep in mind that immediately after elution, you have to neutralize your protein, because otherwise it will also be denatured in glycine. Depending on your downstream application, it might also be an option to use your protein bound to the beads. Like this, you can avoid the problems with elution. Well, after finishing your IP experiment, you normally check by Western blot or Coomassie whether your IP was successful. And if it was successful and you see your protein of interest in the bound plane, everything is fine. But as I mentioned, often two problems occur. So the first problem is that you have a weak or no pull-down. And if you haven't already done it, so please check in your input lane, so load the input lane and check whether your protein is really soluble in your lysis buffer. Because if your protein still is in the pellet, you can't expect to get a nice immunoprecipitation. Then you can also try to prolong the incubation time of the antibody with the lysate. This might especially help if you have a really high volume or if your antigen is very diluted in your lysate. And please also remember that not all antibodies are bound to protein A or G. So check whether your antibody is really bound, because otherwise you again cannot expect to have a successful immunoprecipitation. Well, another problem might be that you have really high background. To reduce your background, you can try to prolong the washing steps. If your protein is stable and also the interaction partners, you can go up to 5 minutes per one washing step. It's no problem. Then you can also try to increase the stringency during the washing step. You can increase the sample volume, so especially if your lysate is very concentrated, this might also help to reduce your background. You can optimize the detergent concentration. And what we also observed, which helps, if you transfer the beads to a new tube after the first wash. Because then all proteins that are bound to your tube will stay in your tube in which you prepared your IP, and you just transfer the protein which are bound to your beads to a new tube, and this protein you can then elute. Well, during the last slide, we discussed many parameters which can influence the success of your IP. And all these parameters you have to optimize again and again for every new antibody. But as I already mentioned before, good antibodies are available against tagged proteins like MYC, HA, FLAG, or GFP. And if it's possible for your application, you might think of using your protein to a protein tag. Then you can be at least sure that the antibody is working and that the antibody is not the problem during your IP. And you can focus on all the other parameters which you might have to optimize. And a really good antibody against a tagged protein is the GFP-trap. The GFP-trap binds with very high affinity to your GFP-Fusion protein. And using GFP as a tag, you also have the advantage that you can easily combine microscopy and biochemistry. But what is now the GFP-trap? So just a small excursus to immunology. Conventional antibodies have a size of about 150 kilodalton and consist of 2 heavy and 2 light chains. Camelids have, besides the conventional antibodies, also antibodies which are so-called heavy chain only antibodies, and they consist only of 2 heavy chains. And the antigen binding domain of these heavy chain antibodies is the smallest known naturally occurring antigen binding fragment. In the case of the GFP-trap, such a small fragment directed against GFP is covalently coupled to the matrix like agarose beads. And this then results in the so-called GFP-trap. And especially the strong binding of the GFP-trap to GFP is a big advantage in optimizing your immunoprecipitation. The GFP-trap was used in thousands of IPs. It's published in more than 500 publications. It's ready to use, so as I said, just take it out of the fridge, wash, and then you can immediately start your immunoprecipitation. And it's available coupled to different matrices like agarose or magnetic beads or even 96-well plates. The GFP-trap will also work in all standard lysis buffers. But even if you have to use a special buffer, the GFP-trap will probably work since it binds so strong to GFP. And the GFP-trap also recognizes the native form of your GFP protein. In the following experiment, GFP was immunoprecipitated from cell lysate using the GFP-trap. And the pull-down efficiency was checked after 1 minute, 5 minutes, 10 minutes, and 30 minutes by Western blot, loading the input, the flow-through, and the bound fraction. And as you see, after 10 minutes, only a minor band is visible in the flow-through lane. And after 30 minutes, all GFP was immunoprecipitated from the lysate, showing that the GFP-trap is not only very specific, but it is also very fast and efficient for the isolation of your GFP-fusion proteins. So to reduce your background in your IP, we mentioned that you might have to wash with very high stringency. Using the GFP-trap, you can increase the salt concentration in your wash buffer up to 2 molar NaCl, or you can even wash with 8 molar urea if it is necessary for your downstream application. Even during such harsh washing conditions, the GFP-trap will bind to GFP. Elution can be done, as we discussed before, with all the standard procedures. So you can elute with SDS sample buffer, or you can elute with glycine pH 2.5, or you can also use the beads directly in your downstream applications. So finally, I will show you a couple of slides of possible downstream applications using the GFP-trap. So probably the easiest and fastest downstream application is the analysis of your IP by Western blot and Kumasi. In this experiment, a pull-down of GFP was compared, so using the GFP-trap or conventional antibodies against GFP. And as you see, GFP was pulled out very efficiently using the GFP-trap. And since the antibody is covalently linked to the beads, you don't have any heavy and light chain contaminations in your Western blot or your Kumasi gel like you normally have it doing your IP with conventional antibodies. Especially the harsh washing conditions can be also a big advantage if you want to do mass spectrometry. But you can also use your beads or your enzyme in enzyme activity measurements, and you can do chip and rip analysis after pulling down your GFP-fusion protein using the GFP-trap. So in summary, we showed you that immunoprecipitation is often not trivial, but don't be scared. So it will work if you use the right tools. And the success of the immunoprecipitation is influenced by many parameters, and as already mentioned many times, especially the choice of the antibody. And to focus on your experiments, you might use established state-of-the-art tools like tag antibodies if it's possible. So now, thanks a lot for your attention. And if you have questions, please write the questions, and we will try to answer them in the Q&A session. But if we can't answer them due to time limit, please visit our homepage or send us an email, and we will contact you within the next 24 hours. And now I will hand over or back to Mike.
Thank you, Tina. Yeah, and just as a reminder, you can submit questions at any time during the presentation, simply using the Q&A box at the bottom of your screen. So let me introduce our next speaker. Our next speaker is Dr. Mike Mortellaro, and he is a co-founder and an owner of Bulldog Bio, a firm that specializes in bringing high-technology products to North American life science researchers. Welcome, Dr. Mortellaro.
For taking time to listen and learn a little about electro-kinetic transfer and how it may relate to your research. At Bulldog Bio, we strive to find useful and interesting products based upon emerging technologies. Many times, This allows us to work hand in hand with researchers as they explore and develop new uses for these products. As you've just seen how very powerful antibody tools from our partner Chromotech can be used to improve upon immuno-purification procedures, we're taking time to listen and learn a little about electro-kinetic transfer and how it may relate to your research. At Bulldog Bio, we strive to find useful and interesting products based upon emerging technologies. Many times, this allows us to work hand in hand with researchers as they explore and develop new uses for these products. As you've just seen how very powerful antibody tools from our partner Chromotech can be used to improve upon immuno-purification procedures, now here you'll see how a new wrinkle on an old technology, electroporation, can vastly improve the ability to move molecules into cells. This technology, electro-kinetic transfer, has been perfected by our partner, Nepogene, out of Chiba, Japan. They have over 20 years' experience in the electroporation market, and by carefully studying the effects of low-voltage electroporation, have patented a new way of using this old technology. First, what is the difference between electroporation and electrokinetic transfer? Electroporation is a simple process whereby living cells are placed between two opposing electrodes and are subjected to an electrical field of short duration and high voltage, referred to as a pulse. The electrical field disrupts the plasma membrane of the cells, temporarily creating holes or pores. This allows molecules present in the cell media to diffuse into the cells before the membrane reseals. There are some features common to electroporation. Typically, electroporation is a single pulse between 300 volts and 3,000 volts in under 5 milliseconds in length. This pulse is exponential in nature and delivers a relatively large amount of energy to the cells. In the case of robust walled cells, such as bacteria, the method is quite effective and can result in high levels of transformation. However, more delicate walled cells in almost every type of animal cell are unable to heal after exposure to such a high jolt of energy. This kills all or nearly all the cells. In recent years, a better way has been developed to alleviate the cell survivability issue while actually boosting delivery efficiency. At the highest level, electrokinetic transfer is simply taking the energy applied in a single pulse electroporation technique and parsing it out over a long period of time and a lower voltage. Looking more closely at the pulsing pattern reveals that there are typically two types of pulses applied. To the right, you'll see the two types of pulses as displayed on an oscilloscope attached to an EPA-21 system. One, referred to as the pouring pulse, simply creates channels in the cell membrane, much like electroporation. The difference being that the voltages used to porate are a magnitude lower than those for electroporation. The second type of pulse is the transfer pulse, and this is used to carry charged molecules from the media into the porated cells. I like to think of it as electrophoresis, but on a much shorter time frame and, of course, in a different matrix. In actual protocol, the pouring pulse phase typically applies 1 to 9 pulses between 50 and 300 volts, each under 50 milliseconds in length. The transfer pulse phase typically has more pulses, 2 up to a few dozen at a much lower voltage, under 50 volts, and a longer duration, greater than 50 milliseconds. I'll stress now that the type of cells, electrode, growth conditions, buffers, and target molecule will impact the optimal pulsing pattern. Nepogene has developed a large library of pulsing parameters linked to many different types of cells, tissues, and organisms. So what can electrokinetic transfer offer? Well, first, electrokinetic transfer accepts a broader range of compatible buffers, mostly due to the much lower voltage being applied. Cell-friendly buffers such as DMEM, OptiMen, PBS, and even seawater are compatible with some systems. Electrokinetic transfer promotes higher cell viability as compared with traditional electroporation, and often better than reagent-based delivery technologies. It works with mammalian cells, live tissue explants, and living organisms. In general, the low voltages and physiological buffers create a happy environment for these living cells. Some of you might find it surprising that electrokinetic transfer can work with a broad range of target molecules. It is not only useful for DNA transfection, but it can be used for RNA knockdown experiments. More impressive is the technology of moving charged molecule is easily applied to proteins or synthetic molecules. These molecules do not need to be small. In fact, recently we've had a lab successfully transfect mammalian cells with an artificial chromosome. The electrokinetic transfer technique is appropriate for many applications and has resulted in an explosion of custom electrodes. We offer over 150 different electrode types, each designed for a specific set of applications. To the best of our ability, we work closely with each of our customers to ensure they choose the best electrode for their application. On a related note, electrokinetic transfer, while powerful, does not always guarantee better results than some of the other related technologies. We typically guarantee transfer efficiencies that is at least equal to other methods. That is coupled with a typically higher survivability in broad range of applications. A webinar is not a great platform for providing precise details for improving electrokinetic transfer experiments. Nevertheless, there are some simple rules that will go a long way to the successful delivery of DNA, RNA, or proteins into cells. First, choose the right electrode. This can sometimes feel daunting, but the literature is a big help in this case, along with the electrode company's expert advice. I'll go through some types of electrodes quite quickly to give you a feel for the breadth of this parameter. Cells in suspension, you'll typically use a cuvette-style electrode, either 2 millimeters or 4 millimeters in gap, and they fit within the chamber on the right-hand side. You can actually do cells in adherence as well. This you would use a handheld electrode that has 3 electrodes coming down. 2 are anode and 1 is cathode, and it's placed directly into either a 35 millimeter dish or a 24 well plate. We have many types of electrodes for ex vivo work, which are tissue explants. Here's one which is our Petri dish with a stick on an electrode. We also have a huge host of electrodes for tissues in vivo. In this example, here's a needle electrode at the bottom. We have many types and styles. On the right-hand side here is a tweezer-style electrode that has both the anode and cathode on top and bottom of this forceps. And you can even get very specific types of electrodes, like this one for skin, which we call the spoon and fork, where you place the spoon above the skin and pierce under the skin with the tines of the fork. Next, it is important that the instrument you use to pulse can maintain a square wave. Once you lose a square wave, you can no longer get repeatable results. It's that simple. Carefully wash your cells just prior to pulsing. In our experience, serum is bad for the cells during the actual electrokinetic transfer process. We suspect that the serum contains molecules that make their way into the cells, causing them to become sick. It is very important that the molecule, whether protein, RNA, or DNA, be as pure as reasonably possible before pulsing. The reason is similar to the serum issue. Lots of things can cause cells to get stressed. Try to minimize these. Everyone wants to minimize the amount of time an experiment takes by multitasking and batch prepping. Do not do this for electrokinetic transfer procedures. The amount of time that cells spend away from a standard media environment directly impacts their survivability. So after each sample is pulsed, immediately replace the media with serum-containing media. For electrokinetic transfer, shortcuts are typically bad. Did I mention shortcuts are bad for electrokinetic transfer experiments? One of the biggest strengths can sometimes be perceived as a weakness. In this case, the open access to controlling many parameters around pulsing provides you with the opportunity to finely tune your experiment. As hundreds of labs in Japan can attest to, if you take advantage of this ability, you'll be able to achieve remarkable results. However, if you're not willing to spend the time to optimize pulsing parameters, then you may never get great results. In general, we hand out a spreadsheet with about 4 to 12 different pulsing parameters depending upon the application. We always stress that all parameter sets should be tested whenever starting a new application or cell type. In general, the pouring pulse is the most important parameter to focus upon, as this is where the balance between enough energy to open pores and too much energy to kill cells is struck. Please note that the pulsing parameters for even the same cell type can vary by lab. We believe that the NEPA-21 is best suited for the multitude of current and future electrokinetic transfer applications. First, the NEPA-21 is the only system to have a non-capacitor design. So what does this mean? Well, it means it maintains square waves even at physiological levels of salt. Most of the experiments our customers perform are between 30 ohms and 100 ohms. As you can see on the right, a non-capacitor design, as shown in panel A, does not have a capacitor to bleed out in low resistance environments. So whether applying 1,000 ohms, 200 ohms, or 50 ohms, the square waves are maintained. Conversely, all other electroporators behave as in panel B, where the square waves quickly morph to waves resembling exponential waves of bacterial systems. Impedance feedback is invaluable for saving you time and money. The NEPA 21 was the first and still one of only 2 systems to offer this important information. We always recommend to measure the impedance of your sample right before pulsing. If it is within the range typical for the experiment, then your results are much more likely to be repeatable. If outside the range, it gives you the chance to troubleshoot and correct the problem before wasting your sample. A completely unique aspect of the NEPA 21 is the ability to reverse the polarity of the pulse. We see that this simple inversion can improve transfections for some cell types by 10 to 30% in efficiency. Though we're not quite sure why, we suspect it has to do with physically moving the target molecules back and forth in solution when you reverse the polarity. What really sets the NEPA 21 apart from electroporation system is its open platform approach. You're not forced to work in a black box. With the NEPA 21, you have the ability to see and control every aspect of the pulsing. This includes the number of pulses, the voltage, the length of time for pulsing, the interval of time between pulses. You can set a decay rate from one pulse to the next if you choose to minimize the voltage exposure. And, of course, you can alter the polarity, as I just mentioned. The system is set up to provide you with 4 full phases of pulsing, 2 pouring phases, and 2 transfer phases. Some parameter sets can last for seconds. So what is the real lab experience of NEPA 21 customers? Certainly, many labs accomplish the more mundane types of transfections that other technologies are capable of. Delivery of plasmid and siRNA into HeLa, COS, 3T3, and similar cells as a SNAP. But how far can you push the NEPA 21? In the case of primary neurons, we consistently have seen remarkable results. The most common procedure is to perform the electro-kinetic transfer after harvesting the neurons, but before plating. The cells are suspended in media and pulsed within an electroporation cuvette. They are immediately plated after pulsing. Though the results vary depending on cell type and technique, it is common to see both cell viability and transfection efficiencies above 50%. In this case, primary neurons from a mouse cerebral cortex were harvested on embryonic day 14 and subjected to pulsing. GFP transfection was both efficient at 70% and stable. The image was taken 9 days after electrokinetic transfer. Some labs prefer to pulse after the neurons have been plated and attached to the dish. In this case, the neurons were prepared from E15 mouse cerebral cortex. The electrokinetic transfer was performed on day 6 after plating within a standard 24-well culture plate and using the NEPA gene adherent cell electrode mentioned previously. Again, the level of transfection efficiency is very nice. Electrokinetic transfer has been successfully applied to stem cells, induced pluripotent stem cells, T cells, and dendritic cells using the NEPA 21. These cells can be very challenging as they tend to be quite finicky. We find the results vary quite a bit depending upon cell type and lab. In general, we consider it a success when both the transfection efficiency and cell viability are above 35%. In this case, human mesenchymal stem cells were pulsed in a 2 mm gap electroporation cuvette. As you can see, the results were very nice, 78% cell survival and about 75% transfection efficiency. Some cell types have shown to be highly resistant to delivery of plasmid DNA. Some of these types have a cell which adds an added barrier to entry. In some cases, you can remove the cell wall, but survivability is an issue. Over the past year, labs in Japan have cleverly applied the NEPA-21 to solve this problem. Electrokinetic transfer has proven to be very effective for this situation. As you can see on the right, diatoms are amenable to transformation by the NEPA-21. The same is true for chlamydomonas as seen below. Both show a remarkable improvement in delivery efficiency as compared to a standard single pulse electroporation technique. As I've touched upon, electrokinetic transfer is not just for DNA. It can work equally well for siRNA and surprisingly is nicely suited for proteins. In this example, our partner Chromotech used a NEPA-21 system to deliver a novel nanobody linked to a fluorophore. The alpaca antibody fragment was specific to PARP and conjugated to ATO48 for determining localization in the cell. They had success using electrokinetic transfer method to deliver this approximately 15 kilodalton protein into CHO cells. As pointed out by the orange arrows in the time-lapse images below, the PARP nanobody ATO48 hybrid molecule will bind to condensed chromatin during interphase and the early phases of cell division. At the 180-minute mark, the chromosomes are lining up, and by 190 minutes, they have been separated. At the 210 mark, the two daughter cells are fully formed, and the PARP nanobody ATO48 is still binding. Chromotech has also been successful with using a lamin nanobody linked to ATO488. Now we've nearly 100 examples of using electrokinetic transfer for tissue explants and in vivo work. So instead of continuing to show you examples, we've invited one of our customers, Dr. Nandan Nerurkar from Harvard Medical University, to discuss his experiments in greater detail. Thank you once again.
Thanks, Mike. And once again, I just want to remind the audience that you can submit questions at any time using the Q&A box on your screen. So Dr. Nandan Nerurkar completed his PhD in mechanical engineering at the University of Pennsylvania, studying the complex physical properties of native and engineered tissues. Since then, Dr. Nerurkar has focused on understanding how genetic and molecular cues regulate physical forces during embryonic development. The overarching goal of Dr. Nerurkar's research is to understand in both a molecular and physical sense how tissues are quote-unquote built in the developing embryo, and how these strategies can then be implemented to develop novel tissue engineering approaches. Welcome, Dr. Nerurkar.
Thanks, Mike, for the introduction. So one of the central questions of developmental biology is how you can go from a disorganized ball of cells to a fully patterned organism. And we've known for some time that the instructions for this transformation to occur are encoded within the genome, and DNA provides all of the instructions necessary for this to happen. However, something that we know much less about is how this code then enacts physical changes by regulating mechanical forces to actually sculpt an embryo, and that's something that we know very little about. So understanding the molecular and genetic factors that control physical forces to form the embryo is the focus of my work, and it's through studying this that I've been able to apply the NEPA gene electroporation system, and I'm going to talk about how that system was optimized for my particular purposes today. So in this endeavor, we rely primarily on the chick embryo as a model system. Shown at left is a 3-day-old chick embryo, which is equivalent to about 2.5 to 3 weeks of development in humans. As illustrated on the sketch at right, you can see that at these earlier stages of development, the chick embryo and the human embryo are essentially indistinguishable. And as such, many developmental processes are actually conserved across these two species. In addition, fertilized chick eggs are relatively cheap and readily available. Most important to the studies in mechanical forces, the chick embryo is actually very accessible. So you can simply cut a hole in the egg, perform some genetic or physical manipulation, and then simply put a piece of tape over that hole, and the embryo continues developing completely normally. In addition, at early stages of development that we're interested in, you can actually take the entire embryo out of the egg and it'll continue to develop normally for up to 2 days. So these are qualities that make embryological experiments possible in the chick embryo that would never be able to do in mice. However, there's one very big limitation, which is that with the exception of a handful of transgenic lines, genetic tools in the chick are extremely limited. It's for this reason that electroporation is a very, very important tool for the study of embryogenesis in chick. So the process of electroporation has been described very nicely in the previous presentation by Mike, and I'll only touch on how this method applies to the chick. So I'll do this through example. Shown at left is a 2-day-old chick embryo between 2 electrodes. And we take in as example the neural tube. This is the cylindrical structure that's shown in the center of the image. The neural tube eventually will go on to form the brain and the spinal cord, but at this stage is simply a hollow tube. So to electroporate the neural tube, you simply inject the DNA solution into the lumen, as shown in the cartoon at right, and then apply an electrical pulse to introduce the plasmid DNA into the cells. So while electroporation of the neural tube is relatively straightforward, the tissue that we're actually interested in is the endoderm. So shown here is a schematic cross-section of the chick embryo at about 2 days of development. And you can see that the ventral surface, which is the lower portion of the image, is where the endoderm is found. And this is a single cell layer that lines the ventral surface of the embryo, and eventually will go on to form the entire respiratory and gastrointestinal tracts. So this is a difficult tissue to electroporate for 3 main reasons. First, because in the egg it's not accessible because the embryo is ventral side down, so when you open it and look at the egg, what you're seeing is actually the dorsal side. Second, the endoderm is open to the yolk, so it's not a contained cavity, which means that when you apply DNA, you have to make sure that it stays there until the electrical pulse can be applied. And the third and most important of these is that the endoderm is a very, very delicate tissue, and this creates particular needs for electroporation. So dealing first with the accessibility, this is actually a straightforward approach, which is evident from nearly a century's worth of embryology work in the chick. So what's shown in these images here is an egg that has been fertilized and incubated for about 4 days of development. You have this rich vascular network, and at the center of that you have the embryo. So the embryo can simply be removed from the surrounding yolk by putting a ring of filter paper onto the membrane of the yolk, cutting around it, and then pulling that filter paper off. And when you pull it off, the embryo is stuck to it, and now you have that embryo to manipulate externally. So we can make use of this with the dish electrodes that Mike discussed briefly in the previous talk, where at the bottom of this well you have a square electrode, and then you apply an additional electrode to the solution in which the embryo is sitting inside of this dish. And by injecting DNA onto the endoderm directly, which we can do now just by flipping the endoderm or flipping the embryo over, allows access directly to electroporate the endoderm. So, and this is shown by the blue in the center panel there. And now shown in the lower right-hand portion is just the electroporation setup that we have in the lab, where we have the Nipogene electroporator, and it's coupled with a stereoscope as well as a microinjector. And so this allows us to perform very targeted injections and electroporations. And by simply using this dish electrode, we can and culturing the embryo outside of the egg, we can eliminate the challenges associated with the inaccessibility of the endoderm in ovo. So the second issue was that, unlike the neural tube, where you have an enclosed region where you can inject the DNA, the endoderm is open because it's on the outside of the embryo. And so we have to deal with the diffusion limitations, where the DNA can be injected near the endoderm, but will immediately go into the surrounding solution. So the solution to this problem came through a combination of trial and error and also interactions with the lead engineer at Nipogene. The first thing we did was to increase to 5% sucrose the DNA solution, which means that now the DNA is denser than PBS, which means that it's going to sink. And second, we increased the concentration of DNA to a fairly high concentration of 5 to 10 micrograms per microliter. And the result is a very thick, viscous solution, which is also limited in its ability to diffuse away from the site of injection. And then, as is common in these types of studies, you additionally add some sort of dye, which just allows you to see where the DNA solution has gone. So Third, and finally, and this is the most important where I'll spend the most time, is that the endoderm is extremely delicate, partly because of how thin it is, and as well as just the early developmental stage at which these manipulations are being done. And what we found through electroporations using our previous electroporation system is that it was quite difficult to get electroporations to work efficiently in the endoderm. Part of the reason for this is shown on the left side, where if the electroporation parameters are too weak, you do get some signal. This is just a GFP under ubiquitous promoter. And while you do get some efficiency of electroporation, what you see in the very center, which is actually the region of cells that we're most interested in, it's quite dim, and this is because the efficiency of electroporation is too low. Now, we can improve that efficiency by increasing the voltage and duration and other parameters and get something such as what's seen on the right, where now you have this very nice bright signal. However, what you'll notice in the lower half of the image, there's this large hole that's formed, and this is something that we see very commonly, that the endoderm actually has a tendency to burn when it's electroporated. So with the previous systems, the problem was that it was very difficult to find a middle ground between getting enough efficiency but also not damaging the tissue. And this is what led us to consider then the NEPA gene system. Among the features that drew us to the system were the high precision over very small time scales, as well as very large voltage scales that are sometimes lacking in other systems, and also just the number of different variables that you can play with in order to ensure that you get really the right amount of voltage and nothing more than that, so that you can get efficient electroporations without damaging the tissue. So what I'm showing here is the optimized parameters that we arrived at through interactions with NEPA gene, where they actually were quite collaborative and helpful in this process. And there's two things to point out. First is that in the pouring pulse, you see that the pulse duration is 0.1 milliseconds, or 100 microseconds. And this is actually quite different from most of the published parameters for electroporation of embryonic tissue, in that it's almost 2 orders of magnitude lower than what's normally used. This is something that with our previous system we would not have been able to do, and it's because of this that the NEPA gene has allowed us to go forward with this project. The second thing is that in addition to having a pouring pulse, you also have a transfer pulse, which means that you can actually get a significant boost in the efficiency and also the subsequent signal that you generate. Finally, you can see here the reading of impedance that we've provided, and this is actually a very useful tool for doing the optimization, because you can get a readout of the setup of your electroporation experiment before actually applying any voltages to the embryo, and this permits you to make sure that really all of the conditions are the same, so that you're only varying the parameter of interest. So these are some examples of successful electroporations that we've been able to do since switching to the NEPA gene system. At the far left is an image of the endoderm of a chick embryo electroporated with a ubiquitous GFP reporter. And you can see clearly visible cell morphologies. And what's worth pointing out is that this is not an antibody stain. This is actually a live sample, and that bright signal is the result of the endogenous signal from the GFP reporter. So the importance of this is that with a live, strong signal like this, we can now do time-lapse imaging and actually directly observe morphogenetic processes. And this is what we're interested in doing. So shown on the right is a cell track projection over the course of time, and what it shows you is that this population of cells is moving from the top of the screen down to the bottom in this very unidirectional fashion. So in addition to just being able to visualize the cells, we can also manipulate the function at the subcellular level of various genes. So this is shown on the next slide here, where we've electroporated in not only GFP to visualize the cells, but also a dominant negative form of the FGF receptor 1, which means that we're eliminating the function of FGF on these particular cells. And what you can see is that this works with a significant efficiency such that we can actually disrupt the movement of these cells simply through the electroporation of this dominant negative. And this really establishes the electroporation with NEPA gene as a very useful tool for our applications. And so if anyone is interested, the work that I've presented just these very few bits of data from is currently in preparation as a manuscript, and I'm happy to discuss that offline via email or any other means. But to conclude, what we found through the optimization using NEPA gene is that the electroporation of chick requires a very, very short duration of pulses due to how delicate the tissue is. Additionally, in addition to being able to actually do this, the NEPA gene provides a tremendous control over the parameters that are necessary for electroporation, and the result is a highly reproducible transfection with great efficiency and viability, and that this has been sufficient for the live imaging and functional studies that we're doing. And with that, I'll just end by acknowledging the labs of Connie Sepko and Cliff Tabin at Harvard Medical School, where this work has been carried out, as well as Atsushi Miyamura, who is the lead engineer at NEPA Gene, who's worked very closely with me on the optimization process. Mike Mortellaro, who you just heard speak, who was very helpful in terms of choosing the right equipment that we wanted to buy. And finally, the work has been funded by a fellowship from the National Institutes of Health. And with that, Mike, I'll turn it back to you for the question and answer session.
Okay, great. Thank you, Nandan. So as Dr. Narkar has indicated, this is now the question and answer session of the webinar. Again, you can submit questions at any time using the Q&A box on the lower portion of your screen. And so there are a couple of questions that are already in here, and I think I will just try to jump in. The first question, Tina, is for you. Are there applications where you'd recommend agarose coupled trap over magnetic bead trap?
Well, yes, it depends on your application. So in principle, both work fine, but the agarose beads have a much higher binding capacity than the magnetic beads. So if you want to combine both, so the advantage of having magnetic beads and a high binding capacity, we now developed a new product, so the GTMA, so it's magnetic agarose, and there you have again a really high binding capacity, and they, as I said, they are magnetic. But it depends on your application, and maybe the person who asked the question can just write me an email and tell me the application, and then I will answer her or him herself.
Okay. All right, great. Thank you. Mike, this next question is for you, and it is, for adherent cells, can we use EKT? Oh, wait, I'm sorry. For adherent cells, we use EKT using cuvettes for suspension cell. You know what? I apologize. Can we use EKT cuvettes for suspension cells while harvesting? Which method is better, electrodes on adherent cells or cuvette-based EKT?
Well, in general, when you're looking at primary cells and looking to transfect primary cells, we do recommend that you use the suspension method. It tends to give higher efficiencies. With that said, you can also use the adherent. So, for example, if you're looking at, let's say, primary neurons that are typically in an adherent state when they're growing, when you harvest them, they'll kind of ball up and you'll have them in a suspended state. And at that point is the time that most labs will do the electrokinetic transfer in a cuvette electrode. You can, however, and then you'll plate after that, and once it's plate, then they'll stick down, and most of the cells will survive. However, if you're working with adherent cells and you want to look at something while the cells are still stuck to the surface, the matrix, you can use that adherent cell electrode that I showed earlier. It does typically require more volume, get up to the level necessary for that electrode to work, and that usually requires quite a bit more DNA. So the downside of that is that the DNA requirements are usually quite a bit higher.
Got it. Okay, thank you. Nandan, the next question is for you. In the developing chick embryo, how do you decide when to use electroporation versus other gene transfer methods?
Right, so this is a very context-specific issue. It depends a lot on the accessibility of the cells and what state they reside in. So electroporation works extremely well when you're dealing with an epithelium, and fortuitously, most mesenchymal cell populations in the embryo have an epithelial origin. So the time frame over which you can do an electroporation depends to some extent on the proliferation rates of the cells. As cells divide, depending on the plasmid you're using, the plasmid, because it's not dividing, will eventually get diluted out. And so if the transition of the cell from an epithelium to a mesenchyme is within the range of a few days of when you have access to do the electroporation, then electroporation is by far the best method to use. When this is not the case, it's often preferred methods to use viruses, sometimes a replication-competent virus such as RCAS or lentiviruses, depending on what your application is. The limitation of the viral approaches is that, particularly with replication-competent viruses, you don't have very precise control over the region that you're targeting because eventually the virus tends to spread everywhere. So in those cases, when it's possible, I think electroporation makes sense. When it's not, and for example, for lineage tracing over multiple days, something like a lentiviral approach would make more sense.
Okay. All right, thank you. Tina, coming back to you, does the GFP-trap recognize modified GFP, so eGFP, for example, or turbo GFP, etc.?
Yes, it does. I mean, it depends on the sequence of the GFP protein, so it will recognize eGFP, for example. It won't recognize turbo GFP probably, but it also recognizes other GFP proteins or even CFP or YFP, since they are pretty similar.
Okay, great. Mike, do you have any experience with myeloid lineage cell electrokinetic transfer, specifically raw cell lines and bone marrow-derived cells?
Yes, we do. We actually mostly derive from nepogene interaction with several hundred labs in Japan. We have an extensive library of cells and functions and expected types of viability and efficiency. So, for example, in this case, question was about bone marrow-derived cells, and raw cells. So we know for raw 264.7 mouse macrophage-like cells, we've had 70% viability and 50% transfection efficiency in one lab. BMBA, mouse bone marrow-derived basophils, we have a viability of 45% and a transfection efficiency of 67%. And even for primary cells, in this case primary mouse bone marrow-derived mast cells, we have viabilities of 64% and transfection efficiencies for 78%. Now again, that is for one lab doing it with their particular cell types. Lab-to-lab variability is something to be expected, but this is what we've seen in the field.
Right. Okay. Thanks. Nandan, a question for you. How long after electroporation is there significant signal or protein activity, and how long does it last?
Right. So this depends to some extent on the type of enhancer and promoter that's used, as well as the cell type. But in, for instance, the endoderm, as well as the developing limb bud in the chicken, for example, or the splanchnic mesoderm, lateral plate mesoderm, these things, typically you can see signal as early as 4 hours after the electroporation, but the signal is typically quite weak, and it becomes maximally strong about 8 hours later. How long that lasts before being diluted out really depends on the just how proliferative the cell population is. So the faster that the cell population is dividing, the less effect you're going to get from that plasmid as time goes on.
Okay, thanks. We only have time for a couple of more questions, but Tina, let's shoot another one your way. And the question is, what is the shortest incubation time with GFP-trap?
Well, the shortest incubation time is, as I showed, so even after 5 minutes you already have a pull-down of GFP from your cell lysate, but we normally recommend to you to do incubation times of at least 30 minutes. But if it's important for your assay, even after 5 minutes you can have a pull-down.
Let's see, maybe we have time for one more question. And hold on a second. Sorry, I'm trying to pull up some of the other questions here. Okay, Tina, we're going to actually stay with you. What could be the explanation of additional bands which we observed on the western blot of GFP-trap in a time course experiment?
Well, I mean, this is a pretty difficult question, or not so easily to answer. I mean, it might happen that the protein is degraded during the time course. So I would need to check the the procedure of the experiment, I think, directly with the person, because if they just take out samples, let's say after 5 minutes or 30 minutes, and the protein is very unstable, then it might happen that the additional bands are degradation.
Okay. Well, unfortunately, that's all the time that we have for questions for today. But I would like to remind all the webinar participants that any questions that we were not able to address here live will be answered by our presenters via email. So if you have any additional questions, feel free to submit them. So we'd like to thank Dr. Tina Romer, Dr. Mike Mortellaro, and Dr. Nandan Nurikar for sharing their knowledge with us today, and also offer a special thank you to Bulldog Bio for sponsoring today's very informative webinar. And also thank you for coming to see the webinar, and we invite you to keep an eye out for future webinars on BioCompare. Thank you all for attending.